Welcome to our 10th anniversary celebration for the Institute for Quantitative and Computational Biosciences. So for those of you who haven't met me, my name is Tracy Johnson, and I have the pleasure of being the Dean of Life Sciences here at UCLA. And I'm also a professor in Life Sciences in the Department of Molecular Cell and Developmental Biology. And during my tenure as dean here, since the fall of 2020, which is kind of amazing, it seems like it's gone so fast. But during that time, one of the most exciting aspects of the job has been having the opportunity to highlight the really incredible research that happens in life sciences in our various institutes and centers and programs. And I'm delighted to see you all here tonight to celebrate one of those institutes. So as many of you, I have no doubt, appreciate, big data and advances in computational sciences have profoundly affected all aspects of life. And it's also affected all academic disciplines, and life sciences has been no exception. This development amounts to what can only be referred to as a revolution. So we're able to ask completely different kind of questions and take completely new approaches to answering those questions than we could even 15 years ago. 20 years ago, computational biosciences occupied about 5% of biomedical research efforts. But now, today, it amounts to about 50%. So this complete change has altered not only the kind of science that we do, but also who's doing science and how we train the next generation of scientists to ask the really exciting and important questions. So to guide this massive transformation here at UCLA, the Institute for Quantitative and Computational Biosciences was established in 2014 by the current director, Dr. Alexander Hoffmann. So QC Bio is a collaboration between UCLA Life Sciences, Physical Sciences, the Health Sciences, and Engineering. It involves faculty and postdocs and graduate students from across the campus and encompasses about 15 different departments, which range from microbiology and immunology to physics and computer science and to pathology in the medical school. So one of the things that you're going to see tonight is you're going to get a sense of the depth of the task at hand in really advancing quantitative and computational biology, and life sciences under the leadership of Dr. Hoffmann has really been on the forefront of this. So what unifies all of these partners is really a mission to expand quantitative and computational research, training, and education. So before Dr. Hoffmann presents some of the advances and the exciting things that are happening in QC Bio, I'd like to first take this opportunity to introduce someone who really needs no introduction, Victoria Sork, to say a few words about the genesis of QC Bio. So Victoria was my predecessor, Dean of Life Sciences, and she recognized the importance of computational biology and had the great judgment to recruit Dr. Hoffmann here to UCLA to be the inaugural leader of the Institute. Dr. Sork is a renowned plant biologist, elected member of the American Academy of Arts and Sciences and the California Academy of Sciences. And she's also a dear friend. So, Victoria, please.
Yes, thank you. I am so happy to be here, and I'm so proud of what this institute has become. When I became dean, everyone, you go in the interviews, Tracy will remember this, and everyone tells you what to do. And everyone told me is we don't have enough computational biologists, we don't have enough bioinformaticians, and every department was wanting to hire. And there are two things that they really wanted. They wanted us to hire more people so that they could have more collaborators. And the handful of bioinformaticians we had at the time said, please, we have so many collaborators and we can't keep up with this. You need to hire more faculty. But they also said we need to find a better way to handle all these faculty that are demanding the need for computation. And that's where Matteo Pellegrini stepped up. He said he would establish a collaboratory, and we did know we had this initiative to figure out how we were going to address the issue. of computational biology at UCLA, and that had to be one element, and no one wanted to wait. So as we were searching and searching for just the right person to be director, Matteo said, stood up, I said, okay, let's start it, but it's got to be part of a new institute, and we're still looking. So we had a blue ribbon committee looking. Jeff Miller was on it, Paul Banerjee was on it, David Eisenberg was on it, the who's who of the smart scientists on campus, recognizing that computational biology was really the next molecular biology of life sciences. So with that, after much discussion and interviews, we met Alex and we knew this was the person. And boy, when you see what he's done, he was. And I couldn't be more proud of everything you've done, more thankful for Matteo, because the two being part of the Institute, and then everything you continue to do, which you'll learn aloud, and I'm only allowed to speak for 2 minutes, so I've already exceeded my time limit. I'm proud of what you've done. Congratulations, and thank you so much. I'm honored to be able to just be here today. Thank you.
Well, thank you so much for those really wonderful words, and thank you for recruiting me, Victoria.
I'll always be happy about it.
And it's been really a privilege to help formulate the mission with so many of my colleagues here at UCLA, and it was really a collaborative effort. And I think the nature of this being an initiative from life sciences that is inclusive of all these other units on campus has been a key characteristic of the Institute. So today I'd like to give you a little overview of our activities, but I think the main thing is, of course, that we're interested in some of the research, and so we have some of my colleagues here who will each be able to give a little snippet of the type of research that they're doing. And so, as already said, this initiative was really was prompted by this dramatic shift in the availability of biological data. And here is a little graph that indicates the cost of sequencing a human genome. When it was first sequenced, it cost $100 million. And something happened right there in 2008 where the price of sequencing a human genome just dropped on an exponential scale to under $10,000 and then $1,000. And so the human genome has 3 billion bases. There's an enormous amount of data in that one experiment. But in addition, there was great foresight from the NIH that all the experimental data was collected in public databases, which meant that those large quantities of data could be reanalyzed and repurposed for many different types of questions. And of course, electronic medical records became available, and relating the two was important. And as we also recognize today is the algorithmic innovations from computer science, what we now call artificial intelligence, really dramatically changed how we are thinking about what we can do with biological data. So being UCLA meant that we were in a unique position where we had top departments and institutes and programs all within 200 yards on a small campus. We are currently right here. This is the medical school, this is engineering, this is Boyer Hall. We are literally every seminar on campus related to the sciences, to engineering, to healthcare, is within 200 yards. That is a unique opportunity. It also meant that a new initiative like that could really be catalyzed across those 200-yard radius. And so we developed a vision for developing an institute that focuses on catalyzing research in computational biosciences, ensuring that we have adequate research training, and then at a later point we realized that education and our undergraduate programs also, that the undergraduates were demanding to be part of the action, and so we responded. And that vision was developed by these faculty, many of whom are here today, actually, in 2014. at the time, interestingly, given that they were in different departments and different schools, many of them actually didn't know of each other. But that process of developing the vision already created a network of faculty. And so let me tell you what we've done with that. We focused on research. We supported seminar series and affinity groups where research trainees can interact closely, that catalyze collaboration. And of course, many of our faculty are developing software packages and mathematical models that are then brought out and used by many others. We also, through the efforts of Dean Sork, were able to create a fantastic new state-of-the-art facility on the 5th floor Boyer Hall. Thank you, Mehdi, for that picture. It's better than the one I took. And Boyer Hall is a 1970 building. It was designed for a very different purpose, but this gut renovation that was made possible at that time in 2016 really was transformed and our ability to do this type of collaborative research, and it's been a wonderful hub. We recruited many faculty, not just into life sciences, but of course all over the campus. So our list of faculty now exceeds exceeds 60. And what I mean by faculty here are all faculty whose primary interest is to develop software and analysis tools for the interpretation of biological data. That all biological scientists are using these software tools or using software. That's a given. But somebody actually has to develop them, and that's these faculty. And so that meant their work contributed to the fact, as Dean Johnson already indicated, is that the fraction of research effort in biosciences has dramatically increased over the last 10 years. And because their work enabled the adoption of those high throughput data collection technologies and then interpreting that data. And a key reason for that is, as mentioned, is the fact that the QC Bio Collaboratory under Matteo Pellegrini's leadership was able to broaden the impact of this revolution by enabling experimentalists. And I described this with some tongue-in-cheek here about QC skills. This is, of course, it's quality control, but it's also quantitative and computational skills. And this is where the science, science is. So let me tell you just a brief word about the collaboratory and why it's such an amazing synergistic effort. It's primarily a postdoc training program. It's a training program for computational biology postdocs to learn how to collaborate and how to teach. And that puts them on the path for an academic career, and they're more competitive in their applications going forward. But of course, who's the beneficiary of the teaching and the collaboration? Well, it's the experimentalists. The experimentalists are able to take the workshops in bioinformatics, learn the skills that they need to analyze their data, and when they have questions, go back to the collaboratory fellow and consult. And so that broadens the research impact of the postdoc and therefore making them more competitive. The classes that are being offered are broad. We support 16 postdocs in collaboration with the medical school. And so every quarter, there are 16 of these 1 week or 3 afternoon workshops, 3 morning workshops. That the simplest one here is workshop 1, Introduction to Unix. That's essentially, how do I connect to the network? But it's also advanced Python and machine learning and very specialized things like a workshop called High C, which has to do with how to interpret chromosome structures. So we'll hear maybe a little bit more on that, but it's had tremendous impact. Secondly, of course, we've expanded the graduate programs, the PhD programs, and we now have a suite of 5 that cover different areas of computational biology. Grace Zhao is here, and she is the director of the Bioinformatics Interdepartmental Program. The Biomath program is in the medical school, and Genetics and Genomics is in the medical school. Over the last few years, Grace has expanded the Bioinformatics IDP to add systems biology and medical informatics, and we're very excited about that. And so that expands the set of talents that our faculty can recruit, and of course powers their research. And then finally, given that we have these grad programs, we need to ensure that we have a talent pipeline actually applying to them. And one of the challenges that really any academic graduate program faces is to ensure that it represents the diversity of the population. And so we wanted to make sure that we have a talent pipeline that's not only broad, but also diverse. And we created an undergraduate summer program called Bruins in Genomics, or Big Summer. And it really was a darling with all the funding agencies, and we were able to receive a number of grants over the years from different national institutes that supported the training of undergraduates, including undergraduates, primarily undergraduates from colleges and universities that don't have that much genomics yet, given that we have the strength at UCLA. That program now is beginning to serve more and more our own students as well. But the trickle-down effect of this is that given that so many of our PhD students and faculty are engaged in mentoring students, undergraduates, we're developing a community of mentors. These are all labs with the names of the undergraduates that they hosted last year. That community of mentors really affects the climate in a very positive way, the academic climate, given that we're all focused on this goal of broadening and diversifying our talent pipeline. So I'd like to thank actually all of these mentors, because it takes a lot of work to do so, and it's always very exciting. Finally, we strengthened and developed in some cases our undergraduate programs, the Computational Systems Biology program, and which at that time when we started was housed in computer science, but I don't think it was receiving the kind of support that it really needed. And Dean Sork stepped up, and I was able to convince her to bring it into life sciences and appoint Nan Savage as the director, and he transformed it such that the number of graduates was very low, and it has become increasingly popular. And this is a projected line here. And I don't know whether we're going to be reaching it, but certainly the student demand is there. The question is whether we can follow up by recruiting enough faculty to teach the classes. And so I'd like to thank Van and Matteo for this. But if those numbers are true, then it is undoubtedly UCLA will be the #1 producer of computational biology talent. You know, those will be the applicants who will be applying to Stanford, to Harvard, to MIT, to UCSF. And they're highly sought after. Secondly, why is there so much interest in this? Well, one reason is that we've solved a problem that many undergraduate institutions are facing, which is that students who are interested in biology, of course, should be taking mathematics in their freshman year, but are typically turned off by the math classes. That's not a good situation when you're trying to grow computational biology. And so Alan Garfinkel initiated this under Dean Sork's leadership to create new classes for freshmen. And this is a textbook that came out after the curriculum development, which completely changed the way we do teach mathematics, and Eric Dietz is here to run the program. Here's just a little statistic about how many students are interested in mathematics before the course or after the course. That's unheard of, that you take a math course and you're more interested in math after you've taken the course, a required course. So we're not only surprised by this, the world is surprised. This has received enormous amounts of attention. These are articles from a variety of different places, in Scientific American, and USA Today, at conferences. Here's Alan Garfinkel, here's Eric Dietz, here's a presentation by Texas A&M and UCLA collaboration on how to teach mathematics, and even Harvard wants to know how to teach mathematics to freshmen. So we're super excited about what we've been able to do here, and we certainly hope to continue this. We've been very grateful also to some of our recent alumni and more distant alumni who've seen what we've done and have begun to support it with a new lecture series of a distinguished lectureship, as well as graduate fellowships and teaching awards and so forth. And so I really look forward to moving this into the future, given that we have this unique opportunity at UCLA where all the disciplines are together. So that concludes my opening remarks. Hopefully they weren't too long. I'd like to now invite our panelists to talk about their research a little bit. And our first panelists are Mehdi Bouhaddou. You can take a seat over here. He's an assistant professor in MIMG, Microbiology, Immunology, Molecular Genetics. We just recruited you about a year and a half ago, which is super exciting, from postdoc at UCSF, PhD in New York, and undergraduate, I think, at San Diego.
Berkeley.
At Berkeley.
Grew up in San Diego.
Yeah. And so he'll tell you a little bit about his research. Jamie Lloyd-Smith, I have a seat over there too, is a professor in ecology, evolutionary biology, and human genetics. Actually precedes me here at UCLA. You were one of the reasons why I decided to come, because you talked me into it. I will be sitting on this panel. I'll be speaking as a third person. And Xia Yang is over here. She's a professor in integrated biology and physiology and molecular and medical pharmacology. You were also here when I interviewed, and you were certainly a reason why I decided to take this job.
Take the
You take the credit. You know it. So with that, let me ask Mehdi to step up to the podium, and I have your first slide right here. And here's the.
Thank you. Amazing. Yeah, thanks for having me. Excited to be here. So yeah, my name is Mehdi Bouhaddou. I'm a professor in the biology, in the microbiology, immunology, and molecular genetics department. And what we're passionate about is rapid response to future pandemics, right? So we want to be able, as soon as a new pathogen arrives on the scene, we want to have a drug ready for people to take. So the current paradigm is that antivirals are developed individually for every virus, right? And this takes a long time because you have to develop the drug to bind to specific parts of that virus. But today I want to present to you a new paradigm where we can, oh gosh, now I turned it off. Okay, where concept of host targeting antivirals. And the idea here is that viruses are what are called obligate intracellular parasites. So they require the host, for example, our cells, human cells, to survive and to replicate. And so the idea is if we can find those host dependency factors, what factors in the host do these viruses need to survive? And if we can find those that overlap between different viruses, then we can target those pathways instead of targeting the virus. And we actually have an arsenal of drugs that have already been developed to target, for human diseases, right, that we already have. So we have a lot of tools at our disposal. And one way that we do this, that I've done this, is by developing what are called virus-host protein-protein interaction maps. So don't get scared by the complexity of this, but these are these really detailed maps of how the virus actually interfaces with our own proteins. And what this depicts is the diamond in the middle is the viral protein, and the circles around are the human proteins. And if there's an edge, that means there's an interaction between them that we've used a technology called mass spectrometry to identify. And what we've done on this map is we've labeled the human proteins orange if there's already a drug known to target that protein, right? So if the virus is potentially depending on that protein, and we can inhibit that protein, maybe then we can interrupt the virus's ability to survive and replicate. So for example, this drug, dabrafenib, is actually used to treat melanoma, but then the idea is, can we repurpose that to treat viral infections? And would it be effective against multiple viruses? Potentially, yes. The other thing we do, if we can isolate the virus, is we infect cells in the lab, and we perform a technology called phosphoproteomics to rapidly identify the kinases that change in response to infection. And kinases are one of the largest class of drug targets for many drugs that have already been developed. So we found that targeting kinases that change during infection is a really effective antiviral strategy. So just as a recap, my lab integrates this technology called mass spectrometry with bioinformatics as part of QC Bio. And we don't just study virology, actually. This technology can apply to many different areas of biology. So one area that we're very excited about is understanding how coral is bleaching, the mechanism of coral bleaching. You've probably gone snorkeling and you see all this coral bleaching, right? So what is the mechanism at that interface between the symbiont and that coral animal cell? We also have a grant to study, we've paired with a firehouse in LA to try to understand why firefighters are getting cancer at an alarming rate. So we're actually working with these firefighters to understand how toxins that they're exposed to might induce carcinogenesis in their cells. So thank you for your time.
Great. Thanks, Mehdi. I think so in the theme of viruses and viral infections, Jamie Lloyd-Smith has really been a leader in that area at a different scale, at the epidemiological scale. And so he'll tell us a little bit about his research.
Well, thank you. It's a real pleasure to talk to you tonight. So I study the emergence of new infections. Most of these come from other animals. And there are millions of viruses out there in other animals, but a grand challenge in our field is to identify those viruses that have potential to cause a pandemic before they cause that pandemic. So epidemiologists sort of identify three key factors that lead to pandemic risk. Animal to human spillover is required for a new virus to enter the human population. But then the real crux of pandemic risk is human to human transmission. A virus has to spread effectively between humans to cause a major outbreak, a pandemic. Then there's also human population immunity. Most new viruses encounter a blank slate in humans. There's no immunity yet, and that is what enables them to spread so widely. At the same time, virologists and immunologists are studying the fundamental biology of what viruses do at levels of molecular, cellular, up to host tissues. All of these things are going on inside individual hosts, but they very much shape the ability of the virus to transmit. So this is all great, but the problem is that these two things tend to happen in completely different worlds. So we have all this knowledge, but no way to put those pieces together. And what I try to do in my research is come up with mathematical and computational models which take the ideas and the knowledge from these different domains and link them together in ways that enable us to make our best integrated statements. So we've made some progress in each of these domains in recent years. We put forward the first integrated model of animal to human spillover, which connects everything from the ecology in the animal reservoir down through the shedding of the virus, its stability in the environment, human behaviors that lead to exposure, and ultimately the chance of infection in a human. And this model actually forms the backbone for a global Lancet commission on the prevention of viral spillover that I'm a member of. And we're using this model as a way to develop strategies to block pandemics before they even get started by preventing spillover events. We've also worked on human population immunity, and we discovered that humans actually have immunity against some avian influenza viruses that have never circulated in the human population. It turns out that your first infection as a child, when you get seasonal flu as a child, gives you a special broader type of immunity that encompasses some other sort of broader members of the influenza family tree. So depending what seasonal flu is circulating when you were born, you pick up immunity which you carry lifelong, which protects you against certain avian influenza viruses. And this explains some previously puzzling patterns in the case data for these viruses. As it turns out, you may have heard about H5N1 influenza in the newspapers these days. It's spreading strangely in cattle. If you're lucky enough to be born before 1968, then you probably already have some protection against H5N1. Go drink that raw milk. I don't mean that. All right, we're also working on human-to-human transmission. And to do this, we're building computational models that take data that virologists are generating in the lab on virus properties, and they make use of insights about the key mechanisms governing viral spread within the body and beyond. These models then generate robust insights and predictions about transmissibility. So for an example, very early in the COVID pandemic, we were able to build a model to work with some data on virus stability, which predicted the potential for that virus to spread through the air and cause super spreading events many months before this became sort of commonly accepted as the way the pandemic took off. So our goal going forward is to develop tools to predict the transmissibility of a novel virus from things you can measure about that virus. So you can measure them before it starts causing a major outbreak. So we do this by using computational models to unpack the transmission process into its constituent steps, and then link each of those steps to data that you can actually measure. So for instance, we think about what enables a virus to spread within the body and to get into the particular tissues where it's able to shed and possibly infect others. We think about the actual airborne shedding process itself, and then the stability of the virus in the environment before it finds another host, so how long it remains infectious. And then we think about how much a new host breathes in, so the dose that is delivered to their tissues, and the likelihood that that gives rise to a new infection, possibly needing to overcome some immunity if that person has some prior immune experience with that virus. So once we build all these submodels and test them out, then we can put all those pieces back together. This is the vision, to build a model which will predict the efficiency of transmission of a novel virus from things you can measure in the lab. So this enables us to, or hopefully will enable us, to assess the pandemic potential of a new virus before it's out and running in the world, and give us a head start on prevention measures and possibly developing vaccines or getting Mehdi to find new drugs for us. Thank you.
That's great. So we've heard a lot now about how viruses interact with host cells, how they infect, and how the immune system, of course, is a key determinant on the transmissibility of viruses and other pathogens. And my research is focused on the immune system and attempting to contribute on developing really a predictive understanding of the efficiency of that immune system. So the immune system, as you know, is a distributed organ, unlike the liver or the kidney or the heart, which are discrete organs. The immune system is distributed, and there are certain centers of the immune system that we call lymph nodes, as well as the spleen and the thymus, but also the bone marrow, of course, plays a key role. So a number of different locations in the body are key aspects of the immune system, which is what makes it so difficult to study. So currently, as you know, probably aware, if you go to your physician for your annual physical, hopefully you do that, you they can measure all kinds of things about you. They can measure your heart function. They can also look at your liver function, your kidney function. But there's actually no test that determines how healthy your immune system is. And this is also in part what is limiting us in developing ways of making the immune system more effective, because we can't even diagnose when it's defective. Of course, there's certain overt diseases of a dysfunctional immune system, typically diseases of immunodeficiency or autoimmunity, and so we have treatments for those. But sometimes the immune system is only subtly defective, and so, for example, when it's defective in surveilling for nascent cancer cells, we now, the cancer treatments have really been revolutionized through making the immune system more reactive or more sensitive through what is called PD-1 blockade. And when there's too much cytokines and cytokine traps, cytokines, then we can use traps or cytokine traps to capture those. But those are just some examples, but there must be many more. So given that it's a distributed organ and then many different cell types have to be coordinated, we focus in my lab on how cells are interacting with each other and how they're interacting with their own functions through how they convey information about the pathogen. And that's signaling. And so, in other words, how do the components of the immune system communicate with each other? Now, we started small. We didn't look at the whole immune system entirely, but we just started with one cell type. And that cell type here is macrophages, the primary responders to an immune threat. And here in this little video I'm showing is actually the function of a particular transcription factor that is responsive to the exposure to a pathogen. And then it goes into the nucleus and controls gene expression. And there's a lot of information. It turns out there's a lot more information in that process than previously thought, such that the nuclear target genes, the nuclear immune genes, actually have information about the type of pathogen that's on the outside of the cell. So there is a signaling code that determines this. We learned how to build mathematical models to describe this process. And so in some sense, we have a virtual macrophage that recapitulates this process. And that information allows us to further extend our understanding to immune cells like B cells that are also controlling their proliferation and their differentiation. And this is a video I'm showing here where B cells are proliferating, again, in a controlled environment in a dish. But I'm able to, I'll show you a simulation of those B cells dividing and dying well, it looks like those videos are out of sync now, but that recapitulates what is seen under the microscope dish. So our simulations now are able to capture the behavior of immune cells and how immune cells control their functions. So those are the some of our advances over the last 10 years. But that's just we're just beginning to look at individual immune cells. Now, of course, we want to understand this in a broader context. And to that end, we're focused on the need to personalize vaccination. Vaccines are very effective at the public health level, but at the individual level, they've lost credibility. Too many people are not satisfied with the level of protection they're getting, and others might also feel like that the vaccines are exacerbating some other immune problems. So we need to personalize the process, and so to that end, we need to be able to assess the health state of the immune system, and then use that information to guide the personalization of vaccine administration. And to that, so on the side of assessing the health state, we are collaborating with clinicians to profile the epigenome of immune cells. We are studying how that controls the stimulus responses of immune cells, and also the vaccine responses. And so these are all high throughput, next-gen sequencing-based technologies that have really revolutionized our ability to profile the functionality of immune cells. And so that information, we hope, will guide how we then, that information will guide the computational simulations of B-cell evolution in response to vaccine administration, and allow us to develop ways of guiding that process to be more efficient. So, in other words, develop and guide the use of precision adjuvants to improve vaccine responses. So with that, that's our grand challenge going forward. So with that, I'd like to welcome Xiaoyang to the, oh, thank you. Xiaoyang to the stage and to talk a little bit about her research on a variety of different human disorders.
Yeah, it's really great to be here, and really fortunate to have witnessed the growth, the development, establishment, and the growth of the QC Bio over the years. And Alex has been really the bond of all the computational people here, and to get us together. So I studied various different diseases, and I feel like I've benefited from a lot of the colleagues here through this journey. So in my field, is this one? So we study a lot of complex diseases. So here is the list of the top 10 leading causes of death, including heart disease, cancer, stroke, diabetes, Alzheimer's disease, etc. So many of these diseases are on the list of our study. And why we call them complex diseases. So for each of these diseases, the past few years, past decade also, we have been discovering a lot of genetic variants. For each disease, there are hundreds, if not thousands of genes that have been discovered to affect each of these diseases. And also there are a lot of environmental factors that the firefighters get exposed to the fire foams that contain forever chemicals, for example, like our plastic bottles, when we drink water, there's bisphenol A or bisphenol, there are bisphenols, a lot of chemicals. A lot of these environmental chemicals are also affecting our health. And also for each of these diseases, they involve a lot of tissues and organs. Not a single disease is involved in one place. Even heart disease is not just the heart, right? It's the liver, it's the fat tissue, and many other organs are affected. So, and then we really have to understand the molecular insights inside each of the cells and tissues. There are many different types of data, from the DNA sequence, the modifications of the DNA sequence, to the gene expression, like 20,000 genes, and the proteins, metabolomics, or even the bacteria living inside and on us. So it's really important to put all these different data together to understand how they interact with each other in individual tissues or cell types, and to understand this kind of, we call them networks, so that we can truly understand how the different risk factors affect these networks to develop individual diseases. So what we really are trying to understand is what are the vulnerable cell types, tissues, and the networks, and the cell-cell tissue-tissue interactions in each of these diseases. And also, since we're talking about genetic risks and environmental risks, do they converge on the similar networks, or do they affect a different part of the networks? And also, we wanted to understand whether these diseases are connected. Actually, there are a lot of epidemiological evidence already support they're connected. For example, diabetes is predisposes people to Alzheimer's disease and heart disease. So there are connections between the diseases, but do they go through this kind of shared networks across diseases? And then also, how can we, if we understand the disease networks, how can we use the network structure to understand, to identify potential therapies and identify potential modifying lifestyle or diets or other things that can be therapeutic or preventative. So over the years, we have been trying to work on this framework on a lot of these complex diseases. So of course, with so much data here, and Alex just introduced, nowadays there are a lot of databases where data are deposited there, and we could leverage a lot of this big data to understand individual diseases. So we need to develop tools to put this data together to understand this kind of a network structure. So over the years, we have been developing tools such as Metromics, which takes this data to understand networks for individual diseases, and we can also compare the networks between diseases to understand connections. Pharmacomics, which utilize this network to identify drugs that can target the same network. And also some additional tools that try to use the more recent technology, the single-cell spatial transcriptomics, to understand cellular level networks. So there are some tools that we have been working on. And then utilizing these tools and other tools developed by others, we have been looking at the genetic factors, focusing on the genetics. There are so many genetic studies about every single disease. We pull all the data together and look for the tissue-specific networks of each disease across the years. And then also on the environmental side, we have been studying different diets, injury, and endocrine disruptors, including the forever chemicals, and bisphenols, etc., and nanoparticles, which have been found to be present almost in everybody's systems, and also other type of agents. I didn't list the other things like exercise, etc. So for each of these environmental factors, we have a lot of data. We can pull the data and try to understand which tissues, which networks are affected by each of these elements. So where do we go from here? So now we have some ecosystem about it with the tools available. We can pull a lot of the data to model individual diseases, understand what kind of networks are affected for each disease, and we also have tools that could help us reposition some existing drugs by matching the disease networks with the drug networks so that we can find the matches. And we wanted to move forward with additional improvement of the method so that we can incorporate additional data. For example, we're developing Metromics 3.0, which can incorporate the bacteria or the microbe data into the framework. We're also building additional resources to keep more cell type specific and spatial type of networks. And then we are also trying to build on the development of pharmacomics so that we can understand toxicants, right? There are so many thousands, tens of thousands of toxicants. There are a lot of data about them as well, so we're curating all that data to build networks to understand the individual toxicants, and also nutriomics, that nutrition is so important for our health. But we don't really fully understand the different supplement, different nutrients, different diets effects, so we try to build this as well to see what kind of nutrient are most useful for what kind of diseases. And we have been also moving into electronic medical records so that if we identify certain drugs that has matching network with certain diseases, but the drugs are not really currently being prescribed for that disease, is there any evidence in the medical record that shows that it might work? Actually, we have examples. Actually, Monty in my lab has a poster on how we utilize this system to identify new drugs for type 1 diabetes, blood sugar control. If you're interested, you can take a look. And with all this ecosystem, we're hoping to really answer those questions that I outlined at the beginning. How do do the genetic and environmental factors affect similar type of disease networks? Actually, we have done some of that comparison. There seems to be strong evidence that the disease networks are converging between the genetic and environmental factors. And also, we can understand the crosstalks between diseases, between tissues, and also the last one, cross model, I didn't talk about it, because when we develop drugs, we mainly use animal models to test them, but we don't really know how much that can transfer into the human. So actually, cross-model comparison is also an important angle. So with that, I'll move to the next one.
Well, that's already a lot of science, but I was wondering whether any questions or comments, either from the panelists or, of course, from the audience, for what we've just heard, maybe connections or questions of clarification. I was actually wondering, given to Jamie's, the immune health status appears to be an important component in the emergence or the susceptibility of a viral infection leading to a pandemic. And you have the ability to merge data from all kinds of different sources. And I'm interested in the immune health status and how to measure that. There must be opportunity for us to leverage your expertise for the purposes of parameterizing your models and making better predictions. Would you like to comment on that?
Is this thing on? Yeah, absolutely. As I said, we dwell at the interface of immunology with virology and epidemiology. And sometimes when you're studying pandemics, life is easy in this way because everybody's a blank slate and the virus is ripped through. However, as we saw in COVID, that was chapter 1, and then chapters 2 through infinity are all about the battle between the virus and the human immune system, and vaccines as well. And as Alex highlighted, vaccines have heterogeneous effects on different people, which I would mostly characterize like they're very strongly tilted towards beneficial effects, but some people are very high responders and are heavily immunized, almost impenetrable once they're vaccinated. Other people don't respond as well, and that's something else that we are studying in my group, trying to understand how the profile of immunity, which is generated by vaccination, leads to how it influences the ongoing epidemiology by allowing the virus to get in or not. In terms of what Xiao's studying, that is a realm that is very daunting to me, so I'm grateful to you for merging and simplifying it. I think any tools which enable us to bring together these great wealths of data, sort of focus them toward particular phenotypes, particular outcomes that we care about, that will both potentially lead to translation immediately, but also it helps set up other more simple-minded scientists like me who want to take those findings and use them as building blocks in other tools to ask questions at different scales. So I think in terms of the translation of knowledge from one set of fields to another, sort of by forming a team, it would potentiate all of us, I hope.
Totally agree. Although we don't focus on the immune system yet, or the viral or the vaccine response, but the network models that we build are cell type specific that include all kinds of immune cells and also other tissues, right? So I think those network models might potentially be helpful in certain senses, and also if you study the vaccine or the viral infection, if we know the molecular impact of those on individual tissue types, and that's also somewhere we can converge with other information we have as well.
We should really fold in Mehdi, who has studied the interactions of the virus with the host, and all the networks that are engaged and molecules that are engaged there.
I'll send you my network.
Sure.
I think there clearly, we just have to get together more often and have events like this so we can discover the connections and build grand challenge projects. But I can't wait actually for the next 3 speakers, and so I'd like to, for the current panelists to take their seats and for the next set of panelists to come up to the panel. And that's Grace Shao, who's all right. Let me do this.
Sorry.
Oh.
There you go. Yeah.
Grace Shao, who is a professor in integrated biology and physiology. She was also on that recruitment committee 11 years ago when I was interviewing for this job. And she subsequently became the director of the Computational Biosciences graduate program, and we look forward to your presentation. Matteo Pellegrini, who also preceded me, was already a senior professor, I think, at the time, and had started the collaboratory, which indicated to me just what is possible at UCLA. And he's a professor in molecular cell and developmental biology, directs the collaboratory, and also is the chair of the Comp Bio major currently. And then Eric Dietz, who is a professor at integrated biology and physiology, and I was just super lucky to be able to recruit him. He is now my neighbor on the floor, on the fifth floor, and he is the director of the life science core in mathematics that I briefly mentioned earlier. So I'd like to have Grace come up first and talk about her research.
Thank you, Alex. It's such a pleasure to be here tonight, and thank you all for coming. So my lab studies RNA, and we use a combination of computational methods and experimental methods, and we really have benefited from the QC Bio Institute, and it creates a very active community for my lab and the trainees especially, and also created a lot of collaborative opportunities for us. So today I'm going to talk about one topic that we are working very hard on, which is toxic RNA in Alzheimer's disease. And so I think I don't need much introduction about RNA after the pandemic. So I would like to start from the central dogma of molecular biology, which says that DNA generates RNA, and then RNA generates protein. So this is a simple framework, but as we all know in research, nowadays this is rather an exception because a lot of different roles of RNA have been discovered, and we all know that RNA is not only the messenger, so it's not only the molecule that guides the production of protein. RNA themselves have a lot of diverse functions. And just following the virus and immunology stories that we just heard, I'd like to point out that this SARS-CoV-2 virus has only an RNA genome, it doesn't have DNA. And the human genome, of course, is a DNA genome. And thanks to the technologies that Alex introduced briefly, the high-throughput sequencing technologies, in recent years, we have learned that for the human genome, about 76% of the genome is transcribed into RNA. And so we have a tremendous amount of RNA molecules produced from the genome. However, among all these RNA molecules, only 2% will code for proteins. So these 2% coding RNA or messenger RNA, so they are messengers from the DNA, and they will generate the proteins that will be very functional in the different parts of the cells and have been the focus of a lot of studies in the past decades or so. However, now we know that the rest of these RNA, 98% of the RNA molecules that we produce in our cells are non-coding. They don't code for proteins. And we have studied these RNA as the community has discovered many different types of RNA molecules, and we know that many of these RNA molecules play very critical functions in the cells, like the tRNAs will help to translate the RNAs into proteins, and there are all other types of small or long RNAs. However, despite this intensive research in the recent years, despite the fact that we know examples of non-coding RNAs that are critical in cellular function, for the majority of RNA molecules that are non-coding, we do not know their functional impact in the cells. And we also learned recently that as we age and in various disease conditions, RNA molecules could accumulate. And these so-called toxic RNAs, when they accumulate to a certain amount, they can be causing disease. And there are examples and well-studied disease types where these toxic RNAs are very critical, and therapeutic approaches are being designed to actually fight against the accumulation of such toxic RNA molecules. So today I'd like to highlight our work related to Alzheimer's disease, because Alzheimer's disease are very detrimental, and it's a devastating disease to the patient and the families. About 7 million Americans are living older than 65 age are living with Alzheimer's disease today, and this number is expected to double in about 30 years. So recently, we analyzed as a big data analysis project thousands of brain samples and their RNA expression patterns using computational methods that our lab developed to study the different RNA species that are found within Alzheimer's disease brains versus the control brains that we could find. And we found accumulation of abnormal RNA species in these data sets that mimic these toxic RNA species in other diseases. And we're trying to develop methods to better predict their function, and also doing experimental validations to see that these toxic RNAs really exist in cell culture models of Alzheimer's disease and in larger disease cohorts. And so these toxic RNA species are showing up from non-coding regions, and also especially from these regions in our genome that are highly repetitive. We have many copies of certain RNAs, and that these RNAs in normal cells, they're suppressed by DNA methylation, but during aging or in disease conditions, they could be activated and regulated in a wrong way, so that we have some consequences of having these toxic RNA. And the goal of our research, of course, is to eventually develop biomarkers for early diagnosis of Alzheimer's disease using these RNA species, and using computational methods to identify effective biomarkers that we can measure readily, hopefully in non-invasive ways. And of course, the treatment strategies, once we identify these novel RNA species, how we can actually modulate them early before the disease symptoms get worse. So these early interventions for disease treatment is also another goal of our research. So that's about my research, and because I have also the other role, so I'm tasked to talk about the graduate program, the Bioinformatics Interdepartmental Program, or IDP, that I'm a director for. So this is a fast-growing graduate program that we started in 2008, and it offers both PhD and MS degrees. And in 2008, we started with the bioinformatics major or home area within this graduate program, and that was the only degree program we offer. And then we expanded to have medical informatics in 2019. That was the first year we recruited our class of students. And then next year, we're going to welcome new students in this new home area called systems biology. So we're growing rapidly. And we receive more than 280 applications each year between the bioinformatics and medical informatics. So we're not having students yet for systems biology. And among these applications, we eventually recruit about 11 students into our program. That's about 3.9% of all applications. So you may be wondering why such a small percentage, and that's limited by funding. So that's why we're actively looking for graduate fellowship opportunities, scholarships, and funding opportunities for these very talented students. So our students engage in cutting-edge research, so they are the engines of these excellent research efforts that you have been seeing today and also elsewhere at UCLA. And there are many different types of research within bioinformatics, computational biology that our students are leading, ranging from machine learning and artificial intelligence, data science to epigenomics, gene regulation, and everything in between. And also, which an aspect that's also very important is that our graduate students also engage in teaching and mentoring of undergrad students in the labs and in the classroom. So they are also the driving force of training the next generations of young scientists and recruiting more students into these PhD training for the future. So that's all I want to talk about. Thank you very much.
Thank you, Grace. I think super exciting work. I think asking Jim Pellegrini to tell us about his research interests.
So my lab does many different things in the realm of computational biology, but one of the recurring themes over the past few years has been the study of aging, and I guess we're all interested in aging at some level or other, especially as we age. So there's sort of a general global interest, and a lot of research funding is going into this area, and ever more so even in the biotechnology and such. But of course, most aging research is focused strictly on humans, and it's hard to study aging in humans because we age so slowly, right? And so imagine doing a study, you have to wait many years to see the outcomes. But of course, this isn't the case across animals. There are some animals that age very quickly. So mice and weasels tend to live only at 1 or 2 years. And then at the opposite end of the spectrum, there's some very long-lived animals like whales that can live a couple hundred years. And so to study aging, we try to, we're molecular biologists at heart, so we look for molecular processes that change as we age. And one of the discoveries that was made here at UCLA about 15 or so years ago was that our DNA changes as we age. And it doesn't change in the traditional sense that it accumulates mutations or the sequence of DNA changes, but it changes in other ways, that it gets modified. And specifically, it gets modified by these molecules called methyl groups that are added to our DNA. And remarkably, the changes in DNA that occur kind of track our lifespan. So they change at a very slow rate, at the same sort of rate at which we age. And so one of the things that we've been interested in is studying this process across species. And we've developed this technology called epigenetic clocks, which are essentially molecular clocks that track DNA methylation. And based on DNA methylation, they can tell you how old the individual is. And this can be done across any species. And one of the things we noted is that if we track our aging based on our methylation, we see that it's not linear. That is, we age very quickly early in life. When we're young, we're children, our bodies are changing, our cells are changing, and our DNA is also changing. And it changes very rapidly. And then as we grow older, it changes ever more slowly. And we have some mathematical equations that can be used to describe these processes. We call these sort of saturating exponential functions. And based on these trajectories that we can measure mathematically, we can infer the lifespan of the species. And what we've discovered then is that by tracking DNA methylation, we can not only just observe aging, but we can also determine what is sort of the intrinsic molecular lifespan of a species. And we can essentially create a mathematical model that predicts what the typical lifespan of each species should be. And what we find is that this model reflects pretty much what's been measured for species lifespans. And for instance, for humans, we find that our intrinsic molecular lifespan is around 80 years. Whereas for mice, it's closer to 1 or 2 years. And then for cats and dogs, it's around 15, 20 years. And so this, we feel, is a sort of a useful molecular tool that now, if we could understand the underlying mechanisms and molecular processes that are regulating these changes, might tell us a lot about whether we can influence aging across species. So basically, what my lab has been studying is the development of these intrinsic molecular clocks across species, and using them to infer the intrinsic lifespan of these species. And ultimately, though, what we want to know is, can these processes be modified? And we've known for many years that there are interventions that, at least in other species, can dramatically change the aging rates, as well as these molecular changes to DNA. One of the most dramatic is caused by reducing caloric intake, especially in the short-lived species. You can increase the lifespan of a mouse by 50% or so if you feed it less. Unfortunately, that's probably not the case for humans, and you just get very hungry and don't live much longer. But maybe that gives us insights as to how we can come up with interesting observations. And so we believe these molecular observations will provide insights and motivation to continue the study of aging and how we can modify the rates of aging. And the other thing I've been doing is leading the Computational and Systems Biology undergraduate program, which you've heard about. So it is the undergraduate program that allows us to train our freshmen and those who enter our program to become experts in this field that you've heard about today, and essentially then go on to continue their role in graduate school or professional. We actually find that the majority of our students will continue their education in graduate school, in master's and PhD programs, and eventually go on to research careers or other types of applications. So the major is very hard. It requires broad training across a wide range of disciplines, physics, math, biology, computing, etc. So it's a truly interdisciplinary approach. And despite this fact that it's a very difficult and challenging major, it's grown tremendously over the past few years, as you keep hearing. There is an interest in students to get into this area because they see it as a promising future professional opportunity. And the program itself is broken down into the same areas that you're hearing about today, either focused on bioinformatics, the study of DNA, the systems biology, the mathematical formulation of our cells, and the other one is biomedical informatics, where we also teach students how to analyze images and health records and things like that.
Thank you very much. Fantastic. No, really fantastic work. So our last speaker today is Eric Dietz, and I think he will introduce himself.
Oh, I'm sorry. Oh,
No.
Yeah, I'm also hungry, but I'm not going to live any longer. It turns out, probably, which sucks. Anyway, I'm Eric. I want to thank Alex for recruiting me. This has been a fantastic change in my career and life moving here. And as I'll tell you later, one of the most amazing things about my career is the ability that I have to work with these incredible UCLA undergraduates in part of our sort of undergraduate, especially first-year freshman math program. But before I talk about that, I'm going to talk about some work we've been doing in the lab, and I will say that this work has been completely catalyzed by the fact that my lab has been here in LA. So one of the things that we're interested in is one of the things I think is the most fascinating fact in biology. Every single person in this room started out as a single cell. All of you, and me too. We were all at one point in our lives just one cell. And obviously now we're not. We're trillions of cells. And somehow those trillions of cells self-organize themselves into me or you, something that can walk around and talk and think about baseball or whatever. And so it's pretty astonishing. And one of the critical aspects of this process is the fact that during development, as we become ourselves, as that single cell divides and grows into the adult animal, they differentiate into a bunch of different things. So I couldn't talk to you right now if I didn't have those cells, muscle cells. Muscle cells have the job of contracting. I couldn't think about my talk if I didn't have neurons. And neurons are obviously absolutely critical, not only for controlling the muscles, but also for giving me thoughts, like I want to eat dinner, or whatever it is, right? So this is absolutely critical. And then we heard a lot about immune cells. So these are an immune cell called neutrophils, which are critical for finding pathogens in our bloodstream and eating them and killing them. So as you can tell, looking at all of these different pictures, muscle cells look a lot different from neurons. And they look a lot different from neutrophils. And it was known in the late, by the moment we had the ability to build microscopes, to actually watch this process and actually see what happens during development, people watched it and they realized that they were starting out with one cell, but producing all of this diversity. So the 1800s, we knew that was happening, but we didn't know how it was possible. And around 40 or 50 years later, people realized that the molecule that contains information inside of living things is DNA. And the astounding thing is that all of these individual different looking cell types have the same exact DNA. And so the question is, how is that possible? How is it possible to generate so much diversity in what these things look like and what they do from just one instruction code, just one set of instructions for protein sequences and things like that, just one genome? And so the answer to that we think about today was promulgated by a person named Conrad Hall Waddington working in the 40s, before we had the structure of DNA, before we knew anything about methylation or any of that stuff. Conrad figured out that what had to be happening, or at least he posited or proposed, that you start off life as a single cell, and then as development goes, which is shown by sort of going down this way in this picture, these cells meet these sort of bifurcating pathways. So he posited the existence of an epigenetic landscape and actually coined that term, something that has to be kind of above or beyond the genome, more than the genome, an epiphenomenon to the genome. And this epigenetic landscape allows these sort of cells, as they move through development, to say, for instance, hit this bifurcation here, and sort of some of them will go this way, some of them will go that way, and the ones that go this way will become, say, neurons, and the ones that go that way will become muscle cells. And the idea here is that the bottoms of these valleys correspond to different types of cells. And you might ask, well, how do they know to be different? What's different about them? Well, the modern answer to that question is given by the central dogma that Grace thankfully talked about earlier. The idea is that obviously these cells need to make proteins, and so the notion that's arisen is that each of these different cell types make different types of proteins. So you have proteins that are typically found in neurons, proteins that are typically found in different muscle cells or whatever, neutrophils, etc. So they should be sort of, what's inside the cell is different. And that makes the way the cell reacts to the environment different. It also allows it to function differently. And those proteins and what have you regulate each other. And so they essentially have developed this cool way of a system that autonomously differentiates into all these different, very stable, very well-defined states. And the idea here is that over the past 10 years or so, we've developed incredible new technologies to actually, using the sequencing technologies and other approaches, to measure the amounts of these molecules inside individual cells. And in particular, we talked about sort of DNA to RNA to protein. We have tons of data where we can measure different levels of RNA in millions of individual cells. So what my lab has done, and this is actually done in part by one of my bioinformatics graduate students, Tim Hamilton, who is here and will give a poster, who did a fantastic analysis that I can only briefly describe. And basically we figured out a way to kind of look for these different sort of valleys in this data. And we basically built a computational pipeline to kind of look for them. And the problem is that when we went looking, we didn't find them at all. So this whole beautiful story that I just told you about the biochemical and molecular basis of development is inconsistent with every data set that we can find. And so that's pretty cool. It's not fun. It's kind of sucks because now we have to do a bunch of stuff, and a lot of people don't believe me about it for obvious reasons. But what I think we have to do moving forward here is one of the issues that in this field of sort of single cell biology, what has happened is that we don't have a ton of really useful data. So for instance, you might say, okay, well, one thing we could do is take cells that Alex tells me are macrophages, and Alex knows macrophages. And he'd be like, these are macrophages, dude. Do something with them. And then give me another thing that's like T cells and characterize what's in those cells. We actually have very few data sets where we know the molecular context of well-defined cell types. So we desperately need more data. We have a desperate need, I think, for more new methods. Obviously, single cell data is extremely informative, but the problem with it is that while we can use it to make reasonable models of different cell types, the fact that the data itself doesn't conform to our paradigm means that they can sometimes get messed up. So we have to develop new computational methods. That's what my lab primarily does. But I would also say we have a desperate need for a new theory for the molecular basis of development. So that's long-term, I think, one of the things that I'm hopeful QC Bio will catalyze. So one of the things we also need for the future of science is new scientists. And the problem with humans is that we die. And maybe Mateo will be successful, but unfortunately, I'm probably not going to make it. So the point is that we need to have people to replace me. And so we've developed, and one of the problems we have is that we have such an amazing group of undergraduate students, but they are not necessarily, as Alex pointed out before, enthused or invigorated by the math classes that they take when they first arrive in college. Oftentimes, calculus is seen as being like the worst thing that a first-year student can go through, right? And Dean Sork commissioned an amazing study years ago at UCLA to sort of ask, okay, what is the primary thing that causes students to leave the life sciences? And we found that one of the major contributors to that decision was calculus. And so Dean Sork took a tremendous leap and decided to generate a completely new class. And working with Alan Garfinkel, who Alex mentioned earlier, developed a class that excites an entire new generation about the role of math and computers in biology. And the way that it works is that rather than sort of focusing on mathematical principles that were in vogue in the 19th century, we actually work and teach students the mathematics that mathematicians use today. And that is primarily focused on modeling in biology. Mathematicians would call this dynamical systems theory, which all of my freshmen at the end of their first quarter of teaching, of me teaching them, they'll know, oh yeah, dynamical systems theory, dude, I know what that is, right? So it's incredible what we've been able to achieve here by teaching students the actual mathematics of the 20th and 21st century. We also have an amazing new class that we've developed on statistics that teaches students the computational tools they need to do statistics in the modern era. And we need to do more to engage these students. So one of the things that I'm passionate about is that, I teach 350 to 400 students at a time. And that's a huge group of kids. And what happens after I teach them is that many of them get super excited about studying more math and biology. And then the question is, where do they go? And we do not have the room in CASB to accommodate all the students who get excited. And we don't have the faculty to teach them in their upper division classes from this perspective. So we need to develop new classes and recruit new people, new faculty to do all of this stuff, to basically teach about AI, its applications, and leverage the expertise students have in modeling and in statistics and in computers, which is unique in the entire country. We also need to expand research for our students and expand our sort of that comp bio major in order to accommodate all of this interest. And I will say that one of the most amazing things about UCLA is that our second-year students have an unbelievable understanding of math in biology. And I think it's a unique thing that really has been sort of the product of all of the amazing leadership that's here today. So I'm really excited for the future, and I'll be done.
Thank you, Eric, for that, and thank you for all you do and keeping us inspired. I think maybe because we're hungry, maybe we'll skip the discussion and leave that over the refreshments. And so I just like to thank you for all of you for coming and presenting, and I reiterate again just what privilege it is to be able to help guide this communal effort of computational biology at UCLA, and I hope that we are able to do this type of work in the future. You can see that the research is primed to make big discoveries, and with ongoing support both from the federal government as well as our interested UCLA friends, I think we'll be able to be successful. Of course, the vision for UCLA is multiple. For the QC Bio is multiple, both at the level of developing further infrastructure in Bio-Hall, as well as supporting our postdoctoral fellows and graduate students, but also our undergraduates and their research opportunities. And so with that, I'd like to thank you, and if you are interested to hear more about that, please find us. We will be at the reception and enjoying the refreshments. Thank you very much.